everyone, this is Gayatri Prem Kumar over here, and today we are going to see about the CBSE biology practical tips. So since the examinations are nearing, you all have a lot of questions that is running in your mind. So what kind of questions will be coming to you, how to attend the examination, and how to manage the time, very important, because you will be doing your exam as well as you have to show the result to the examiner. So keeping all these things in mind, there are 5 important tips that you have to follow how to write your biology practical examination and get full 30 out of 30. The first thing what we are going to see is the syllabus, and the second thing is the blueprint, that is the mark allocation for each of the practicals that you're going to do, and third one is the preparation of material. So you will be doing the experiment and you need to know, so what is the materials required, what are the chemicals that are prepared in the preparation that you have over there. And the fourth one is important one is the time management, as I told already. So you'll be given only 3-hour duration. So in this 3-hour duration, you have to do the practicals, you have to show the result to the examiner, and at the same time you have to identify the 7 spotters given to you, and viva will be it will be conducted side by side. So keeping all these things in mind, how you're going to manage your biology practical examination and get full 30 out of 30, and these are the tips that has to be followed. So let us get into the video. So out of the 5 important things, the first one we are going to see is the list of experiments that is present in the CBSE board practical. The first one what we have is the list of experiments. In that you have 3 experiments to be done. The one is the major experiment. So what is the first one, major experiment? It is isolation of DNA from the plant material. So you all know that only one major experiment, and that is the isolation of DNA from plant material. The second one is the minor experiment. So you have 2 minor experiments present. One is the population density and the other one is the population frequency. So you'll be getting either population density or population frequency. You'll be getting only one out of these 2 experiments for the minor experiment. And the third category is the slide preparation. So again, for the slide preparation you have 2 experiments present. One is the onion root tip mitosis or the pollen germination. So again, in these 2 slide preparation you will be getting any one, either the onion root tip mitosis or the pollen germination. So overall, when you are getting the question paper, you will get isolation of DNA, because only one major experiment is there, that is confirmed experiment. But whereas in case of minor, you will get either density, population density, or the population frequency. Again, for slide preparation, you will get either onion root tip or pollen germination. Sometimes if more number of students are there in the batch, what they will do is they will allocate it accordingly. So this is the list of experiments, 3 different experiments. Now coming back to the spotters. So you have a number 7 spotters. So 7 different spotters are available. In that 7 different spotters, the examiner might decide any kind of spotters. That could be either the slides, they will keep the preserved, or the permanent slides can be kept, or pedigree chart, the pedigree chart showing the autosomal or sex-linked dominant or recessive character. So any one will be shown as the pedigree chart. So all these categories, out of this, any 7 will be shown for your spotters. Then coming to the seed showing Mendelian inheritance, again the seed showing the round color, the shapes. So based on that, how you differentiate. So that will be the next category. Or it could be any disease causing organism. Either they will keep the bottled specimen containing ascaris, or the preserved specimen containing the plasmodium, or it could be any other disease causing organism, entamoeba, even entamoeba can be kept. So any kind of disease causing organisms can be kept as a spotter. So all these are this or that category. And then it could be controlled pollination. Again, coming to controlled pollination, emasculation or bagging, where the setup is kept, and you have to find out what kind of setup, whether it is an emasculation or bagging, you have to find out in that case. And then coming to the pollinating agent. So you will be again shown with the flashcards or any other kind of pictures that indicate the pollinating agent through wind or insect or birds, and you have to find out, you have to write what is the pollinating agent, and then you have to write the description for that. And then coming to the homologous and analogous organ. So again showing the pictures of homologous and analogous organs of plants and animals, even that can be kept as a spotter. And the last one is a model specimen showing the symbiotic association between the root and the leguminous plant. root of leguminous plant and the other organism. So that kind of picture, nodule formation seen in the symbiotic association of leguminous plant. So that can be shown as a flashcard, or any kind of bottle specimen can be shown. So all these, out of this, any 7 can be kept as the spotter. So this is about the list of experiments that you're going to do the writing part. The blueprint for each of the experiment, what we are going to do. So first major experiment that carries 5 mark. So the only major experiment that is present for your examination carries 5 mark, and then comes to the minor experiment. So out of this minor, as I told you already, it could be either population density or population frequency, and any one you will be getting, and you will be getting 4 marks. 4 marks will be allocated for this minor experiment. And then comes the slide preparation. Again, for the slide preparation, you will be getting 5 mark for that. So you will be getting 5 mark for the slide preparation. And you have split up for that 5 mark, 4 mark, like aim, materials required, preparation, procedure, and other details. Then coming back to the spotters. So spotters carry 7 mark. So you'll be getting 7 spotters. 7 ones are 7, and you'll be getting 7 spotters. And each spotter, so out of these spotters, you'll be getting any one of the spotters. So for example, it could be the slide, as I told you, either the slide preparation or pedigree chart, or any one of these spotters will be getting. How to write the spotters? So you have to write as identification and comments. So whichever spotter is given, it will be labeled as A, B, C, D, E, F, and G. So you have to write the given spotter is identified as whatever spotter you identify, whether it is a slide, that could be mitosis, meiosis of grasshopper testes, or whatsoever. So whatever the slide that you're identifying, or TS of blastula or mammalian ovary or whatsoever, you have to write the given test, the given slide is identified as, you have to write what you have identified, and you have to write the comments or description for what you have identified. And if possible, diagram, especially for the slide what you're observing, because it is not possible to draw diagrams for most of the spotters. So it is possible for the most of the time they will be expecting for the disease causing organism or any other slide preparation. But anyhow, for the preparation, you have to prepare the diagrams for all the spotters, but it varies during the examination point of time. So you just be ready with all the diagrams, and during the examination time, based upon the spotters what you're identifying, you can draw the diagrams accordingly. So this is the mark allocation. So my major 5, minor 4, slide preparation 5, and spotter 7, together 21 marks. So remaining 9 marks is allocated for the record work and the project what you're doing. So when I'm talking about the record work, the biology record notebook is there, no? So whatever the record notebook you're doing, that writing part, your diagrams, for all that, along with that, viva questions will be asked. So biology record will be 4 marks, and the project what you're doing, the spiral binding one what you're doing, 5 marks. So 9. So 21 plus 9, so you'll be getting total 30 marks. So we have discussed the experiments, mark allocation, blueprint, and then coming back to the materials preparation. So what are the materials that will be available over there and how you have to prepare? So we'll be seeing that part. And the next one will be managing the time. So how to manage your writing part as well as the experiment, and finally how to attend the viva. Now we will discuss the evaluation scheme for the practical examination. The first category is the major experiments. When I'm talking about the major experiment, we have only one major experiment, that is the isolation of DNA, and this carries 5 marks. And then coming back to the minor experiment, and again for the minor experiment you have either this or that, that is either the population density or population frequency. So you'll be getting any one out of these two categories, which again carries 4 marks. And the third category what you're going to write in your practical examination is the slide preparation. Again, this slide preparation is either pollen tube germination or onion root tip mitosis. So any one you will be getting as the slide preparation category, and that carries 5 marks. And the next category is the spotters. So any 7 spotters will be kept for identification. That could be under any of the category. That could be either in the form of slides, like preserved slide that carries the mitosis, meiosis of mammalian testes or ovary, and then the experimental setup that carries emasculation or bagging, and that setup could be kept, or it could be the preserved specimens in case of ascaris or root nodules of leguminous plant. So any of the preserved specimens can be kept. And again the flashcard showing the homologous or analogous organs of plants or animals, and even the pedigree chart that shows the autosomal or the sex-linked dominant or recessive characters. So any one of these category would be kept as spotter, and 7 spotters will be kept. You have to identify all these 7 spotters, give the identification, description or comments for the following one. And then coming to the record work. So you'll be doing 2 records. One will be the practical experiments what you have done, so that you will be writing as the in your biology record notebook along with these spotters, and the other one is the project record, whatever the investigatory or whatever the project you are doing, that will be the project record notebook. And the mark allocated for the practicals record notebook is 4, and for project will be 5, and viva questions will be asked along with these project work and the practical work, that also include your experimental setup. Spotters and questions will be asked along with these experiments. We'll discuss about the preparation of materials that is used in the laboratory in their experiment. So for which experiments do we require the materials? We require the materials for the pollen tube germination. So you should know what are the materials required and what is the composition it is required, and why each material, in which way it is helpful in promoting the purpose. For example, in pollen tube germination, if you see, we use the nutrient solution. Okay, so in nutrient solution, what are the components that are present in the nutrient solution to make up the nutrient solution? For example, we add boric acid, and then we had magnesium sulfate, and then we add sucrose. So you should know why these components are added, what is the composition of these components in the nutrient solution, and what role does each component help in promoting the pollen tube germination. So you should know these preparation of material which will be helpful in your viva questions. In case of onion root tip mitosis, we use fixative, and then we use a different stain. For example, we use acetocarmine. So you should know what is the purpose of adding acetocarmine. Why we are not adding safranin? Because safranin is a general stain which stains the overall cell, but acetocarmine stain only the nuclear part of the root cell, and that is why we are using acetocarmine. So you should know what is the purpose of using each ingredient in your experiment. And the next category is about the isolation of DNA. If you see the next category of isolation of DNA, so in isolation of DNA, we use liquid salt detergent. So you should know what is the purpose of adding this salt and then the detergent. So why we are adding these things which promote the isolation of DNA from plant material, since these things will help you in your viva questions. So next category, so we have discussed already about the content or the material list of experiments for your practical, and the next category is the mark allocation or evaluation scheme or the blueprint. What is the mark allocation overall? And third category is about the preparation of material, and the next one is about the time management, how to manage your time, and then do your practicals and show the result to the examiner. And the last category is about the project record work and viva questions.